Welcome back, everyone. Next, we have Rakovina Therapeutics Inc. It trades on the TSXV under the symbol RKV and is focused on the development of new cancer treatments based on novel DNA damage response technologies. The company has established a pipeline of novel DNA damage response inhibitors with the goal of advancing one or more drug candidates into human clinical trials and obtaining marketing approval for new cancer therapeutics from Health Canada, the United States, FDA, and similar international regulatory agencies. Please welcome its CFO, David Hyman, and Executive Chairman, Jeffrey Bacha. Welcome, gentlemen.
Thank you, Anna. It's a pleasure to be here.
All right. We look forward to hearing about your company. Take it away.
Very good. I trust everybody can see the slides. We founded Rakovina Therapeutics in the spring of 2021 through a CPC transaction, which listed us on the Toronto Venture Exchange. And as Anna mentioned, we are focused in the cancer therapy space. Brief agenda, we'll talk a little bit about the company, our research programs. I'll turn it over to David for some of the financial highlights, and then we'll move on to some Q&A. So when we founded Rakovina Therapeutics, this is based on our team's vast experience across many years in developing drugs in the cancer field. We wanted to put together a company that had a diverse portfolio, so multiple shots on goal, which of course helps to manage the risk of drug development. We have a team around the table that has been doing whatever they're supposed to do in our time and in their roles for many years. We've established a highly efficient research collaboration with the University of British Columbia, which we will talk about. And we've been presenting our progress along the way in peer-reviewed scientific meetings. We'll talk a little bit about how important that is to us in terms of communicating our progress along the way. And we also believe that our valuation is very attractive for an entry point for investors. And we've got a great shareholder base that's been very supportive along the way. We've all seen these kind of slides before where, as we make progress, we expect to create significant value for shareholders. In our experience, this is very real in life sciences and pharmaceutical development. In my experience, personally, taking a company from a stage similar to Rakovina Therapeutics and moving it through milestones, including establishing and launching clinical trials and forming pharmaceutical research partnerships, can take a company from the valuation similar to Rakovina to several $100 million in a relatively short period of time. And we anticipate hitting those kind of milestones over the next couple of years. So cancer as a background is still a very important area of research and focus. It's touched all of our lives at the company, and I'm sure many of your lives in the audience today. We've done a lot of good work across the industry in developing new treatments, but what we have to keep in mind is that cancer is a smart disease. The cells in a tumor come from our own cells. They have the full complement of survival mechanisms, and their job is to survive. So as we develop new and better treatments, that cancer cell is trying to evolve and become resistant to those treatments. So our job is to stay one step ahead of that and continue to innovate and continue to benefit patients as we go forward. And the area of innovation that Rakovina Therapeutics is focused on is DNA damage response. This has become a very important area of research in the cancer field over the last several years. There are 4 drugs on the market, which we would consider first-generation DNA damage response inhibitors. But now that these have been on the market, we are beginning to understand their limitations, and the research, including ours, has shifted toward the development of next-generation products. So DNA damage response inhibitors have become very important in the treatment of breast cancer, prostate, ovarian cancer. These are major cancer markets. These products are marketed, the current ones, by the major pharmaceutical companies that we've all become familiar with through the COVID pandemic. These are the companies that are marketing the vaccines that you hear about, so Pfizer, Glaxo, AstraZeneca. And early in the life cycle of the DNA damage response field, there were several major acquisitions because those big companies knew that these were going to become very major products. So Pfizer bought Medivation, GSK bought Tesaro. in the last couple of years, what we've seen is that that investment has shifted into earlier stage partnerships as companies, including Rakovina Therapeutics, have begun to research next generation technologies. And the reason that we are researching next generation is because we've begun to understand the limitations of these very important first generation drugs. So our portfolio consists of 3 distinct product candidates, all covered by separate patents, all being researched in parallel, that come at different angles of the limitations of the very important first generation products. Our KT2000 series recognizes that the first generation products are not particularly great at getting into the central nervous system. They can't really cross the blood-brain barrier at a therapeutic level. And so what happens when you have a tumor that metastasizes to the brain, which happens in about 50% of breast cancer patients, for example, that drug cannot chase the tumor there. So the KT2000 series is being designed to address that. The KT3000 and 4000 series are what we would consider smart molecules. They have a dual function. The 3000 series is designed to overcome resistance to treatment. So when a patient receives one of these first generation drugs, they will initially respond to treatment, but over time that cancer is going to become resistant to therapy and come back. And when it comes back, it grows very quickly. So we need to be able to address that. And the 3000 series is designed to do that. The 4000 series is actually designed to expand the field, take these types of drugs into areas where they're not available for treatment today. So just a little bit of a background on the DNA damage response and the whole reason we do what we do. So the field is built upon the fact that there are systems within our cells that are designed to detect and repair damage to our DNA. And this happens constantly. It goes on in the background. We don't know what's happening, but it's critical. Every day, tens of thousands of times a day, we're experiencing toxins or UV light or just natural mistakes that happen as our DNA is replicated as a cell divides, become 1, goes to 2, goes to 4, et cetera. So these systems are designed to detect and repair that damage that happens naturally. What we've learned in the last few years is that certain cancers will take advantage of a defect in one or more of those systems to allow a tumor to form. And here's a little bit of a cartoon about this concept. So in a normal cell on the left here, the happy blue cell, you have an A and a B. And this is generally intended to depict redundancy in these systems because they're so important to our cells and to our survival that we'll have a primary and a backup system. What happens on the right here in the example is that you can imagine a defect in pathway A. Well, when that happens, that allows a tumor to form. And if that, eventually that patient is going to be diagnosed with cancer. But when that happens, that repair pathway that's critical for the cell's survival is no longer available to that cancer cell. And that cell becomes entirely reliant on pathway B. So the drugs we're developing, and all of the field is based on this concept of synthetic lethality, target pathway B. A normal cell doesn't notice the drug is there. So we end up with a much safer, targeted therapy that's really designed on a more personal level to attack that tumor. A lot of people are familiar with this concept without knowing it because of the actress Angelina Jolie, who had a dual mastectomy because she was concerned about developing breast cancer. The reason that she was concerned about it is in her history and her family, they have what's called a BRCA mutation, which is a fairly common gene that leads to the formation of certain types of breast cancer. That is a defect in pathway A in a DNA damage repair system. So the drugs that are first generation PARP inhibitors is what they're called. That's actually targeting pathway B in that setting. So this is the concept that we build the company around. Our KT2000 series, as we mentioned, is designed to cross the blood-brain barrier. The first generation drugs that are on the market that generated a combined $3 billion in sales in 2021 and have become standard of care in those BRCA mutated cancers, those cancers that I described with a defect in pathway A. What we now know is that there's some challenges with side effects, and they're also not very good at crossing the blood-brain barrier. And when a cancer metastasizes to the brain, we have the inability to treat that patient with these current drugs. So now this field is shifting, including our research, into having safer products, which have the ability to cross the blood-brain barrier. And our portfolio, we have a number of candidates that we're screening. We believe this is going to be a very partnerable program early in its life cycle. And as an example of this, a PARP1 specific molecule that is purported to cross the blood-brain barrier. Early clinical trials, Merck KGaA put up $65 million just a couple of months ago to get access to that product. So we believe that this program with a PARP1 specific, safer molecule that has good brain penetration will be very attractive to major pharmaceutical companies in terms of partnering. Our KT-3000 series, as I mentioned, is a bifunctional or smart molecule that's designed to overcome treatment resistance. And we'll show you a little bit of data about what we're talking about here. Along the way, we've been presenting data at peer-reviewed scientific meetings. On the left side of the slide are some of the major cancer meetings which we've been invited to present data. And also here is the St. Baldrick's Foundation, which is a major funding agency which saw our data and got behind this program. So we're very pleased to have them on board with us. So here's a little bit of data. So what happens in the cell when you're knocking out pathway B? In a cell that has a defect in pathway A, that cancer cell wants to survive. And so what it's going to do is going to try to turn on pathway A. It's going to mutate and reinstate the function of that original pathway. What we know is that there's a target called HDAC, which is involved in that process. And so the idea is to inhibit the PARP functionality, pathway B, and inhibit the cell from being able to turn back on pathway A. And what these data here are showing is that we have drugs that can do exactly that. Now, early on, when people were thinking about this concept and understanding this mechanism of resistance, everybody said, well, we've got HDAC inhibitors on the market. We've got these 4 first-generation drugs on the market. Let's just combine them and treat the patient with both. On paper, that's a great idea. In practicality, that's had some challenges in terms of toxicity, where you can't really deliver the drugs at the same time. So our chemists design molecules that are very potent at both targets. And the table at the top here is demonstrating that, where you can see at the PARP target, this target knocking out pathway B in the system is very potent and similar to a multi-billion-dollar drug from AstraZeneca. On the HDAC line, you see that AstraZeneca drug has no activity. It was never designed to. Our drugs are very similar to a Merck drug that is on the market that inhibits HDAC. So we have these dual functional molecules, and the idea is that by combining them in these 2 activities in one molecule, you avoid doubling down on the toxicity and will be able to treat the patient safely. At the bottom here, what we're showing is what happens in the cells. And these are data that we have presented at major peer-reviewed meetings. What happens on the top here, where you have the BRCA mutated situation, your defect in pathway A, the AstraZeneca molecule has a potency of about 30 nanomolar. We're about 10 times more potent against that cancer cell, which is exactly what the data from the table above would predict. What happens when we go into that cell and turn that gene on, you'll see that the AstraZeneca number blows up to 2400. That means it stopped working. And you'll see that our drug, by inhibiting that HDAC pathway, inhibiting the turning on of pathway A, maintains its activity against that cancer. And these are the data that have gotten, I think, a lot of folks in the scientific community excited. We recently published a paper around this program, and this is the program that's most likely to lead to our first clinical candidate and initiation of clinical trials. All of our work is being done in collaboration with the University of British Columbia. This is a unique collaboration that allows us to have an in-house dedicated research team in the laboratory of Professor Maz Dugard, who's the president and chief scientific officer of Rakovina Therapeutics. This also gives us access to state-of-the-art facilities. It's very efficient in terms of being able to move quickly in our research, but also in terms of cost, because it's a fixed cost relationship. So our burn rate is very, very predictable. And this has been a great collaboration for us. So as we have moved forward, we are currently optimizing lead candidates for advancement into human clinical trials. When we started out, we had in vitro proof of concept. We moved forward and selected the most promising molecules, and those have now moved on to in vivo toxicology and pharmacology studies. And over the next year or so, we anticipate moving initial programs into IND filing to allow for the initiation of human clinical trials. and to establish some strong partnering opportunities, we believe, particularly around the KT2000 series. And these are the types of milestones that we showed at the beginning that really lead to that potential hockey stick valuation and growth. We have been presenting data at peer-reviewed scientific meetings. These data are all available publicly. And we found over the years of doing this that the best way to communicate what is frankly relatively complicated science to a broad audience is to go through a peer-reviewed setting. That means that we're not just putting out a press release saying we did something interesting in the lab, but rather we are having a vetting process with experts in the field who have reviewed the data in advance and said, these data are important enough, novel, and they should be showcased to the regulatory community, to the medical community, and the potential pharmaceutical partnering community. As I mentioned, the team around Rakovina Therapeutics has a tremendous amount of experience. I won't go through this in detail, but you can see on the right-hand side of the slide, there's big numbers. Everybody's doing whatever their role is in terms of the advisory capacity, whether it's scientific advisory or board of directors or senior management. Everybody's been in that role for 20 plus years. And we've, in most cases, all worked together in the past with some great successes. So David, I'm going to turn it over to you to walk through a couple of financial slides.
Yeah, thanks, Jeff. First of all, some select financial results. We have a calendar year end, and we recently reported our third quarter results, which are on the screen here. I won't go through the items line by line, but highlight that we exited September with approximately 1.4 million in working capital, which translates into about 8 months of runway based on our current burn rate of $170,000 per month. As Jeff mentioned, we have a very predictable cash flow stream, or expenditure stream, due to our fixed contract relationship with UBC, which gives us great visibility on our upcoming needs. In terms of capitalization, we currently have 70 million rounded shares outstanding. There are still some in escrow, about 13.8 million, as you can see on the screen, giving us a public float of about just under 56 million shares. The shares in escrow are held by a private company that's controlled by Jeff and our board of director, Dennis Brown, who control those shares, highlighting that the escrow shares initially held by the CPC group that we did our transaction with to go public have all been released at this point. In terms of dilutives, oh, sorry, Jeff. Very, very clean capital structure, very few dilutives outstanding. As you can see, most of the instruments are priced at or near current market levels. All the incentive options are held by management and directors who are incented, of course, to create value at the same level as new investors would be at these prices. The agent warrants were issued in conjunction with our go public transaction, and the warrants, as you can see, that are with a 40-cent strike price, expire this coming March.
So as we mentioned, the company is traded in the Toronto Venture Exchange under the symbol RKV. This is a bit of our trading history, and you'll see that it's been a challenging year in the markets, but we're pleased with the being able to what we would call hold the line, as there have been some of the cheaper original CPC shares that from the public listing of the CPC have come into the market. We've been blessed with a very supportive group of shareholders. Those original CPC founders, as well as others that have been supporting us, have really helped to keep the company, in terms of the valuation, solid over the last year as the markets have been challenging, and I think really position us very well for growth as we go forward and hit milestones. According to Yahoo Finance, about 45% of the shares are held by insiders. Those are the SETI filers, but I would venture to say that the actual number of closely held shares is significantly higher than that, based on our understanding of others in the market. Recent highlights include our presentations at peer-reviewed scientific meetings, the St. Baldrick's Foundation support, and also the achievement of several patents as we've gone forward over the past year. So really, this last slide, same as the first, this is a company that we're very excited about, based on the fact that we've put together multiple shots on goal in a very rapidly emerging and very important field of cancer research. We've got a team that's done this before. The relationship with UBC is positioned to let us move very quickly toward selecting the right molecules to move into clinical trials. We've been seeing great peer recognition in terms of the invitations and acceptance of peer-reviewed scientific meetings, and a recent publication that we released online, as well as great backing from a very supportive group of shareholders that we're very lucky to have around our table, and really position us to be able to move forward and continue to grow the company. We have, I think, some very attractive milestones ahead of us that we think will not only position us to have products that are going to have great benefit for the cancer patients who we are targeting, but also will unlock significant value for shareholders as we move into human clinical trials and look to establish significant pharmaceutical partnerships. So thank you very much, and we'll turn it over for questions at this point.
Great job, gentlemen. All right, so we have a question from Tyler Watts. So he says you're burning about $500K a quarter, and you have about 1.3 million in cash. So talk a little bit about that. Are you capitalized sufficiently to support the burn?
So when we started the company, if you go back and look at all the documentation and everything we said we would have capital that would be sufficient to move us through several years of lead optimization, and being able to select a molecule to move forward into clinical trials. And we've also said that we would need to raise additional capital to support the launch of clinical trials. So as we go forward, looking out at opportunities to raise capital to support clinical trials, ideally at an accretive valuation, we've been working with our existing shareholders to make sure that we'll have the opportunity to raise that capital at the right time. But for now, we have the runway in front of us to hit the milestones that we've described.
Tyler Watts also wants to know if you have an estimate as to when sales will begin, or is a buyout of the technology the plan?
So most companies like Rakovina Therapeutics do get acquired along the way. You can look at that in history, and I'd say that is a fair likelihood. But our job is to keep our eye on the ball and move the products forward into clinical trials and toward commercialization, and make sure that we're capitalized to do so. So, it's likely that somebody will come along and acquire the company. It's also likely that we'll be able to establish partnerships, which will provide non-dilutive funding to support the programs and commercialization through partnerships as well. So the timing from where we are today to achieving sales revenue is several years away, but the opportunities for achieving partnerships and significant value growth through that are much more close at hand.
Eileen Roberts wants to know if the KT 2000, 3000, and 4000 share some of the same patents, or are they all unique?
They're actually all separate patents. So the chemistries behind them are all unique, and they've been designed to do different things. But while they are unique and different, what we have an advantage of is that in the early stage of our research, there's a tremendous synergy in the lead optimization steps that we're taking. So we can actually run all of those in parallel, which helps us tremendously in terms of being able to be efficient and keep those shots on goal moving forward together. Eventually, they will diverge into different clinical targets, and they are, as I said, unique patents behind them.
Darlene J says, you mentioned Pfizer invested $65 million. Are they focused on what you're doing?
So Pfizer acquired Medivation in order to get access, in part in order to get access to the PARP inhibitor that they are selling. Yes, all of the big pharma companies have programs in DNA damage response. And in particular, AstraZeneca has a major program, but all of the major pharmas recognize that this is a key area of research, and understanding DNA damage response and developing products that exploit that understanding is really going to be beneficial for cancer patients. Those first generation drugs on the market, while they have limitations, have been game changing. And we all know that. And the process of any industry is you have a first generation that changes the game, and then you build upon that, whether it's in pharmaceuticals or cell phones. And that's exactly the process that we expect to see in the DNA damage response field, and we're very excited to be part of it.
Mark Mitchell wants to know if you believe, he wants to know about your competitive advantage versus other small cap companies in the same sector.
So when we put together a company, and this is just our general philosophy, the three things that are critical are people, products and technology, and capitalization and finance. So we start with the people, and if you review our team, everybody's been doing this for a long time, and there's many successes under the belt of people. In fact, one of our advisors, Len Post, was the architect of the Medivation drug that led to the Pfizer acquisition. And so, great people around the table who have done this before, including in this field. In terms of products, we started with three distinct candidates in terms of the three classes of molecules. They all do what they're supposed to in the cell, and it's nice to have multiple shots on goal. So we started from a strong standpoint, and we've rapidly moved forward. And from a finance perspective, we raised the initial capital to let us hit the milestones that we needed to over the first couple of years. And we're blessed with a very strong group of supportive shareholders who we believe will be in a great position to help us with the next round of financing. So, this is, people, products, financing, getting that right, I think positions us with a competitive advantage versus peers.
Fantastic. Mark also wants to know about your decision to list the company on the TSXV. Why? So
the Toronto Venture Exchange has been, a great source of capital for young companies across many industries for many years. I've been familiar with it and the CPC process. And it's a great way for young companies to be capitalized and be part of the public markets, which gives you access to additional capital from a broad audience. So it's really those reasons are, the reputation of the exchange and the history of success, as well as access to capital. And it's been a great position for Rakovina Therapeutics to be in as a young company, and we look forward to building from there.
And last question, Mark wants to know about your relationship with the University of British Columbia. What are some of the advantages you see from that relationship?
Well, this is somewhat of a unique relationship. I've worked with UBC over the years, great institution and very commercial friendly in terms of being supportive of the community and biotech around British Columbia in particular. A lot of great technology has come out of there. These technologies didn't come from UBC, but we worked with Mads de Gaard in terms of our due diligence on some of the assets that we were bringing together. And Professor de Gaard got so excited that he said, "Hey, can we do this in my lab? How can I be part of the company?" And so we approached UBC about making that happen, and we worked it out so we could have a dedicated team in his lab and build the company from there. It's given us access not only to great infrastructure in terms of that laboratory, but also access to animal facilities and toxicology and all the tools that lead optimization requires. So rather than having to send our drug candidates out to a third-party contract research organization, which is very time intensive, it takes a long time to get that research back. It is very expensive. We can do this all in real time, and that's really helped us to accelerate our development and be in the position that we are today. And it's also a very cost-efficient relationship, as we mentioned. It's a fixed cost arrangement, which keeps that burn rate very predictable. If you go back quarter over quarter in our financial filings from inception, it's pretty steady, and that's a great position to be in.
Wonderful. Well, thank you, gentlemen. Do you have any closing remarks for us?
I really appreciate everybody's time this morning. Please feel free to reach out. We're very excited about the year ahead and the opportunities to move toward and into human clinical trials, as well as hitting other milestones related to pharmaceutical partnering. Feel free to reach out, and we look forward to sharing our progress going forward.
Wonderful. Thank you so much for joining us, and please come back soon with some updates.
Thank you.
All right. Thanks, Anna.
All right, everyone. Stay with us. We're going to transition to our next presenter. We'll be right back.